Welcome, everybody, to the ninth ACCRF patient webinar on antibody drug conjugates in clinical trials. My name is Jeff Kaufman. I'm the Executive Director of ACCRF, and I'm joined today by ACCRF Director of Research, Dr. Nicole Spardy Burr, Nikki, as well as by ACCRF's Communications and Development Officer, Reed Moats. We want to thank you for taking part of your weekend to join us, and we sincerely hope that this is educational and hopefully inspirational for you. The agenda for today, I will begin with me giving overviews of first the foundation, then about systemic therapy generally, and about clinical trials generally in ACC. And then I'll hand things over to Nikki, who'll talk about the more pressing issues of clinical trials for ACC and antibody drug conjugates, or ADCs. These ADCs are a promising new modality for therapy, and we anticipate that they'll keep being important for patients for the years to come, and that's why we want to educate patients about ADCs. We'll finish up with a question and answer session moderated by Reed, and the questions will come from those that you all submitted when you registered for this webinar. But before we begin, I just have to go through a few housekeeping items. If we could go to the next slide. We will be monitoring the chat for questions, and we'll try to address as many of them as possible within the hour that we have. We ask that you not put any personal information into the chat or share anything from the chat. Now the legal disclaimer, this webinar does not represent medical advice. ACCRF seeks to educate patients so they may discuss treatment decisions with an experienced, licensed physician in a private setting. We simply don't have the information on your background for your particular case to ever be able to provide medical advice to an individual. We're trying to educate generally about the treatment options available. And finally, this webinar is being recorded, and we hope to post it on the website, hopefully within the next couple of days, so it will be available for reviewing. So now let me start with an overview of the foundation. If we could go to the next slide. There we go. So my wife Marnie and I started ACCRF nearly 20 years ago. It'll be 20 years in December, in fact. And what you see here is a picture of our family the week before Marnie went in for surgery on her ACC tumor in her right parotid salivary gland. And you can see we had 4 young boys. It was a hectic time, a scary time. But we were fortunate that we found other like-minded patients, who really wanted to push for better treatments for our community, and who had the same urgency to find those treatments. And that's why we started the foundation. We were also fortunate to have a group of some of the world's leading oncologists to join our scientific advisory board, which has guided our research agenda over the 20 years. Next slide, please. Our mission is very simple. We want to accelerate improved therapies and a cure for ACC. We believe that the best way to do that is to develop a pipeline of clinical trials based on the best available science that demonstrate benefits for patients. We want to help others generate the data and the evidence that can make patients feel more comfortable in choosing the treatment options for them. Next slide, please. ACCRF sits in the middle of an ecosystem with 4 major components. First and most important are patients. Nothing happens in research without patients. There are no specimens, there's no funding, there's nobody to go in clinical trials. Everything begins and ends with patients. We also have very strong relationships with academic institutions. More than 50 academic medical centers are part of our network, and it's a very global enterprise. We have universities and institutes across the entire world involved in ACC research. We work closely to co-fund grants and collaborate with other public and charitable funders like the National Institute of Health and other charities. And we also have very close relationships with private industry. These are the companies that discover and develop and eventually provide the drugs that we hope will lead to better outcomes for ACC patients. We are unlike a lot of foundations in that we actually have our own ACC preclinical models, and these serve as a very important way to attract companies to look at ACC and to consider making new drugs for ACC patients. And so that we're often involved in testing whether new drugs may be appropriate for ACC patients. Next slide, please. The last 20 years have been, an amazing journey. The landscape of ACC research has changed dramatically since 2005. We've raised about $28 million, given nearly 200 grants. Our grantees have identified the key genetic drivers of ACC. On prior calls, you may have heard webinars, you may have heard of the genes MIB and MIBL1 and Notch. So we have a much better understanding of the biology of ACC. The number of scientific articles on ACC is much, much higher. The preclinical models are available. More than 150 drugs have been screened in those models. And unlike in 2005, when there was, I believe, just one clinical trial that was available to ACC patients, we have about 20 trials open currently that might be appropriate for some ACC patients. I'll end by saying that we do not have an approved drug for advanced patients with advanced disease. However, there are two drugs that are now mentioned in the NCCN and ASCO guidelines as the best available drugs for advanced ACC. And we hope that we will be seeing some FDA and EMA and Japan FDA approvals in the coming years. And that's what we're working towards. Next slide, please. Now, you're on this webinar because you want to be informed. I think possibly the best way is visit our website. We have a lot of great resources on there with a physician's list, second opinions page, pages that talk about open and completed clinical trials. And there you can sign up for our quarterly newsletter, where we'll continually update you on new research, new trials, and patient events. Or feel free to email us directly. We are trying to expand our social media presence. And at the bottom there, you can see we do have a presence in Facebook, Instagram, LinkedIn, and YouTube. So feel free to follow us on any of those platforms. Next slide, please. So let me start giving context to this entire webinar, which is really about systemic therapy. And the ADCs and clinical trials are about trying systemic therapy. I think the first point to be made is that almost all patients have surgery and radiation, and many of those patients actually are cured by surgery and radiation. They get local control where the primary tumor was. And so many patients go through a period where they have no evident disease. Those patients may be done. There may be no need for additional treatments such as we'll be discussing today. Now, the problem is we don't know which patients are cured. There can be recurrences after many, many years. And so it's important that patients have lifelong or at least very, very long follow-up and monitoring to keep track of their ACC. The second point is, this webinar is not about those patients who don't have NED or no evident disease. It's about patients with recurrence, current cancer that is returned either to the primary site, and that's a local recurrence, or a tumor that's traveled to a different site in the body, and that's a metastasis. And what I want to emphasize is that, patients should stay positive and hopeful. Some patients may never have a recurrence. Some of you on this call, on this webinar, uh, may never have that, and that's maybe about half of patients is our guesstimate. Many patients who do get recurrences can have stable or very slow-growing metastases for many years or even decades. So, that's another scenario. And even those patients who do get recurrence and they are growing, there are some options to stabilize the disease, at the moment. And we're working on better options, and that's what this webinar is about, is to inform you about some of those. Next slide, please. So let me talk about some of the key questions in systemic therapy. First of all, what is systemic therapy? And systemic therapy are drugs. It's either drugs that you take orally, in your mouth, or that are infused into the veins of a patient. And the key thing is that systemic therapy flows throughout the entire body, and that's the entire system. And that's as opposed to local therapy. And local therapy might be surgery or it might be radiation, where the treatment is focused on just one part of the body. So that's what systemic therapy is. Uh, when do you consider a treatment for a recurrence? And in speaking to many of our, most expert oncologists, often they will say that they won't even consider a treatment for a recurrence until there's at least one measurable, 1 centimeter recurrence that is progressing, that's growing 20% or more over the past 6 months. So that doesn't mean that you have to get treatment at that point, but typically most oncologists won't even consider treatment until that point. And how do you choose, when it is time to get treatment? How do you choose between a local therapy such as surgery or radiation versus systemic therapy? The fact is there is no great roadmap. It's not perfectly clear and laid out what patients in that situation should do. The treatments can depend on how many tumors there are, how large they are, where they're located, how much they're growing. And these are topics that need to be discussed with an experienced ACC physician, preferably as part of a multidisciplinary tumor board, where you've got surgeons, radiation oncologists, and medical oncologists who all discuss the case. Typically, systemic therapy is going to be the most appropriate treatment type if there's very widespread disease. If there certainly, if there's like 20 metastases, but typically it's if there's more than 5 tumors, you might at least consider systemic therapy. And the question is, which drug should a patient choose? As I've said before, there are no approved drugs for advanced ACC. The clinical guidelines from NCCN, ASCO, and ESMO, they all point to the first best option being a clinical trial if it's available. And if it's not available, then to try the best available drugs. And what are those best available drugs? Currently, they are TKIs, or tyrosine kinase inhibitors, like lenvatinib or axitinib. And these drugs, they are not a cure, but in many patients, they can stable the disease for more than 6 months, sometimes years. Unfortunately, they do cause side effects like high blood pressure, fatigue, and nausea, and they can be difficult to take for long periods. I know that sometimes you hear that chemotherapy doesn't work in ACC, and that may be somewhat the case with slower growing ACC, but it is the case that chemotherapy is widely used, especially for faster growing tumors, and especially in places where there may not be access to some of the TKIs. Next slide, please. So now let me turn a little bit to some general comments about clinical trials. And there are studies to test new therapies for safety and effectiveness. And what's really important is that these trials uh, have a consistent way of selecting patients and collecting data. And, I'm all for uh, anecdotes of patients who have done well on a treatment. That is great. But we believe that the way that you're going to make patients most comfortable trying a treatment is to consistently determine, well, what exactly was the situation of the patient before they got on a treatment, and how much of the treatment did they take, and did their tumor shrink 5% or 50%? What you need is consistent um, data and evidence that's collected, and that's what clinical trials do. They make sure that an anecdote is more than an anecdote, that it really gives people an understanding of what happened across how many people precisely. Um, I point out that for patients who do go into clinical trials, there's always the freedom to withdraw. You can choose to leave the study at any time for absolutely any reason. So that's, no one should ever feel trapped into a trial. There are generally three phases. The phase one is a small study that may be, 30 to 40 patients. And there, the goal is to check for safety, to find the best dose, and to see what the side effects are at different dose levels. And typically, the doses start very, very low and then gradually rise as safety is established. And that's called dose escalation. The phases 2 and 3 are larger studies, and they're basically at the dose that was found in phase 1 to have the best balance of effectiveness and safety. And so then these are just larger trials at a dose that should be better and should be hopefully working for patients and hopefully tolerable. But that's what the study is there to determine, is that new dose effective and tolerable. Now, let me go back, make a comment about early phase 1 trials. I talked about dose escalation, and often they start at very low doses, which you wouldn't expect to have a therapeutic benefit. And so on ACCRF's Open Studies website, we historically have not put early phase 1 studies on there, because if you're a patient that needs to be treated, you want to be on a clinical trial that has a chance of actually having a therapeutic benefit. So we would often leave those off until the doses got to levels that we thought would be, or the company thought would be effective for patients. There's been a new development over the last few years, something called intra-patient dose escalation. And that's where a patient who's, historically would have just gone on at a low dose and then gone off the trial. Instead, they stay on the trial. And as there's data that shows that the higher doses are safe, then that patient who starts at a low dose can keep rising to therapeutic doses. And so we have started putting some phase 1 studies onto our website where there is the possibility of a patient staying on and getting to a higher dose. So that's one important new trend that we've seen over the last few years. Another important trend is really driven by precision medicine, which is getting the right drug to the right patient at the right dose. And that's what's driving what that's driving is increased stratification of patients, and that it's segmentation. It's saying, well, this group acts a little bit different than that group. And we've seen that in ACC, where there's been a gravitation towards um, a grouping of patients into two groups, ACC1 and ACC2. ACC1 is the more aggressive uh, um, ACC, where they tend to get recurrences more quickly, and when they come, they tend to grow faster. And those are associated with notch gene mutations, solid histology, and the lack of p63 gene expression. And so then if there are those factors, it doesn't mean you're absolutely going to have a aggressive ACC, but the chances rise that you would have more aggressive ACC. And for ACC2, which is the less aggressive ACC, it can still be very, very serious, but it might take longer till that becomes serious. And there tend not to be any notch mutations and no solid histology. And there's a lot of this marker called p63. And so what you're seeing is a trend towards some clinical trials targeted at, say, just ACC1 or just ACC2. And so there, if you look at our Open Studies website, we'll have a separate section for ACC1 patients below the all-comers section. And it's important to understand that. But also, even if a patient, if a clinical trial is accepting all patients, all ACC patients, as still most clinical trials do, they're keeping track of how many patients are ACC1 versus 2, so we can understand better how to interpret the results. Does a drug work better in one group than the other, or maybe it works exactly the same? Okay. Next slide, please. Well, so this is where, hopefully I've given you a good overview of the foundation of systemic therapy and clinical trials. Now you get to the important part of the presentation where I hand it over to our Director of Research, Nikki, to go over clinical trials and antibody drug conjugates. Take it away, Nikki.
Okay. Thanks, Jeff, and thank you all for being here today. I'm going to start by reviewing some of the clinical trials that are currently open to ACC patients with actively growing either local recurrence or distant metastasis, as Jeff outlined. This table is a little bit busy, so I'm going to walk our way through it. We have 12 trials listed here, but you can always find a more updated list on our Open Studies webpage, and I'll ask somebody from the ACCRF staff to put that in the chat. This page is constantly updated with new trials. Maybe a trial is expanding in terms of the sites or those that are closing. So we have the drug name, the drug modality, which I'll go through in the next slide, the drug target, and that's really, what is the cellular target, the protein inside the cell that the drug is designed to attack or inhibit or get rid of. You have the sponsor, and that's who funded the trial. That could be a company, or that could be a company in combination with a hospital. The phase of the trial, which Jeff went through, these are all phase 1 or phase 2, so these are early studies. The inclusion criteria, which Jeff started to talk about, but this typically could be a patient's characteristic or a condition that really allows them to participate in the trial. So that could be a biomarker, like we talked about with ACC1, or it could be something like a patient may not be allowed to have been pre-treated with a similar type of drug that hits like a similar target. And then we have the sites where the trials are open. If they've listed in multiple sites in USA, you just see USA. And then if it's at a single site, you have USA with the hospital in parentheses. All right, so we're going to start by focusing on the different drug modalities because I think that provides a good context for the rest of the conversation. All right, so the first type of drug I want to talk about are small molecules. So these are tiny chemical compounds that can easily enter the cell. Small molecules are typically designed to bind a protein that sends signals to the cancer cell to grow and survive. These proteins can be found anywhere inside the cell. And the small molecule is designed to bind the protein at a specific location that when it binds there, it turns the activity of that protein off, or it sort of blocks its important function in the cell. And typically, right, these are activities that, again, are telling the cell, grow, survive. I think of this a lot like coming into a room, turning off a light switch, and putting a piece of tape over the light switch. You can't turn the light back on in the room. And so because the cancer cell really depends on this protein and its activity to give it those signals, turning off the light switch is one way that that cancer cell will really feel those effects and hopefully will stop growing or die. The second type of drug I want to talk about are monoclonal antibodies, and these are a special type of protein that's made by our immune system that can only bind to very specific other proteins. And the analogy I like to think of is, your house key only fits your front door. It doesn't fit any other door on your street. So monoclonal antibodies for drug development are typically designed to target proteins that sit on the cell surface. So here you can see it docking right there. And I think of this, a lot of times these proteins are are found at very high levels in the cancer cell because the cancer cell has had a change in the DNA that tells it to make really high levels of this protein. Because again, this protein often is providing those growth signals. So the monoclonal antibody binds here, blocks the activity, and again, because that cell really depends on it, that's a way to target that pathway, turn it down, and hopefully the cancer cell feels those negative effects. Okay, the next section are bispecific antibodies. So these are researcher engineered antibodies so that now you can bind two different proteins. And so the version here I'm showing, it can be two different proteins on the same cell, but here one portion, the pink portion of the antibody is binding to the cancer cell. The purple portion of the antibody is actually binding to an immune cell. And so this is kind of a way for within the body to drag immune cells closer to your cancer cells. And you're hopefully they're trying to increase the chance that the immune system will recognize the cancer cell as foreign and get rid of it. And then this last section are antibody drug conjugates, or ADCs. We're going to talk more about this, but ADCs have your antibody, and linked to them are an anti-cancer drug with the goal of specifically delivering your anti-cancer drugs to cancer cells that have that target of interest. Okay. So now let's walk our way again back through the trials now that we've kind of have ourselves oriented. So we'll start with the small molecules. Okay. And I'm going to focus on the drug target and any inclusion criteria here. So the first couple trials are small molecules that are targeting the MIB protein. And we've heard a little bit about MIB. We believe most ACC tumors have high levels of MIB, and that MIB is sending these signals to tell the cell to grow and survive. So these drugs are really interesting because instead of actually binding to MIB itself inside the cell, they bind some of the genomic instructions that are responsible for making MIB inside of an ACC cell. And when the drugs bind, they force the cell to make short inactive forms of MIB that when the cell sees it, it recognizes it kind of as junk because it's too small to really do anything that MIB would normally do. And it delivers it to the garbage disposal system in the cell. So sometimes these will be called MIB degraders because it's getting rid of that MIB protein. And because we think MIB is so important for, a majority of ACC tumors, we're really excited about these trials. The next drug is called abemaciclib. So this is a small molecule that targets a protein called CDK4/6. And this is a protein that's really actively involved in DNA replication. This trial is only open to ACC type 1 patients. And Jeff I'll just reinforce that this is some combination of patients that have tumors with solid histology, Notch mutations, low P63, or aggressive disease. And this trial actually flowed from some work that our grantees presented at the ACCRF Research Conference. Okay, So the next bucket were these bispecific molecules, and the drug is called ivonescimab. It targets two pathways, VEGFR and PD-1. VEGFR is a pathway that's also targeted by drugs like lenvatinib and axitinib, and PD-1 is part of the immune system. So with this molecule, you're trying to target two pathways at once. Here, the inclusion criteria is all ACC patients, but they can have been pretreated with some type of VEGFR inhibitor. Okay, and then this last, we have several different ADC trials that are open, and we're going to go through those trials once I've spent some time educating everybody on what are ADCs. Okay, so we've already learned a little bit about the antibody, but just again, they're a special protein that recognizes other proteins found on the cell. We're going to be talking about proteins that are on the cell surface, and again, thinking of it as like a key sliding into a lock. You have the payload. This is a drug that causes cells to either stop growing or die. So you can think of these, like chemotherapy drugs that interfere with DNA replication. And so that's the time in the cell's life cycle where it's making a copy of all of its DNA, or during mitosis. And this is when, after cells have made their copy, they start to pull apart from each other, the mother and the daughter. And so these are two phases of the cell's life that are where it's really vulnerable. And so that's why chemotherapy drugs are really targeted at hitting those important processes. And then you have the linker. This is the molecule that connects the antibody and the payload. Okay, so researchers need to think smartly about which cell surface targets would make the best ADC. And so I'm showing you a normal cell here and a cancer cell, and you can see that some cell surface proteins are just on normal cells, like the pink here, or the blue. Some targets are shared across the normal and the cancer cell, like the purple and the green, and some targets are only expressed on the cancer cell, like the orange. And so these are the targets where we want to design our ADCs to kind of dock into, right? With the goal that we can reduce side effects by only directing the ADC to a cancer cell, but leaving the normal cell alone. Okay, so once the ADC binds at the receptor, it's internalized by the cell, so brought inside. And once it's inside the cell, the ligands are either the linkers are either broken apart or dissolved. And this allows the chemotherapy to go directly to the nucleus, where it can do some damage. And if too much mutations or too much damage happens inside the DNA, the cancer cell will say, okay, there's too many mistakes here. I'm not able to fix it. And then this is where the cell, may die or stop growing. All right, so of course we want to ask, what are the ADC targets in ACC? And the work I'll be talking about here has all been done by Dr. Renata Ferrarotto at MD Anderson Cancer Center. She's a longtime ACCRF grantee, and she treats many recurrent metastatic ACC patients at MD Anderson. So she set out to test, and find what are the ADC targets in ACC. And so she had a group of a little more than 50 ACC tumors. And she set out to really look at their molecular fingerprints. That's what I like to call it. So she did DNA, RNA, and protein. So that's sort of different stages and how proteins are made inside the cell. And you can do different tests to ask, what does the DNA look like? What does the protein look like? And so I like to think of that as your molecular fingerprint. Which genes are up, which genes are down? And so then you have this set of 50 fingerprints. And the question is, do they all look the same? Do they all look different? Or can you start to see similarities in your bucket of 50 tumors? And that's what she saw, is that these tumors start to sort of divide into two groups in terms of their fingerprint, right? And that's where we got this ACC1, ACC2 molecular subtype. Now, the subtypes share some things. So, for example, they both have high levels of MIB, but they do have differences. So ACC type 2 tumors, excuse me, have cribriform tubular histology, So they have mixtures of cells, myoepithelial cells and epithelial cells. Myoepithelial cells same positive for P63. That's why P63 still stays high there. And they also express a number or have higher levels of a number of cellular targets that are listed here. ACC type 1 tumors typically have solid histology, like we said. They've lost those myoepithelial cells, so they're lower in P63. They tend to have notch mutations, high levels of this protein called MYC, and MYC is involved in a number of other cancers, and it again sends those signals to survive and grow, and high levels of a protein that's on the cell surface called B7H4. Now, it was really interesting at this time because we knew sort of from the growing literature that companies were developing B7H4 ADCs. So she went on to test this finding in a couple of different types of tests, because these molecular fingerprints are sort of if you really take a high level approach. But now you want to go in with a magnifying glass with different tests and really prove to ourselves ACC type tumors seem to have more B7H4 protein. And so I'll focus on this little picture here on the bottom. So this is a type of test where you're looking at a tumor slide, and anywhere on the tumor slide where you have B7H4, the cell will turn brown. The more brown you see, the more B7H4 you see. And she showed really nicely that ACC type 1 tumors stain very positive for B7H4, so they have high levels, whereas ACC type 2 is much lower. All right, so this set us up to really start to answer the question, can we kill ACC type 1 tumor cells with the B7H4 ADC? Right, so her work kind of showed this. So this represents a collaboration between Dr. Ferrarotto at MD Anderson with us, ACCRF's drug screening platform. So we have disease models that we have up and running that are open to companies, to academics. And the goal there, right, is to bring in drugs and test them in our disease models and see whether the drug is able to shrink tumors in the disease model. And also a collaboration with a large pharmaceutical company called AstraZeneca, which was developing a B7H4 ADC. And so that molecule is called AZD8205. So you can see from our picture now, the ADC has targets to B7H4, and it brings its anti-cancer drug along. Right, so this is a little bit of the data that we worked together, which was then published. So the first question is, Renata showed in primary tumor samples, so tumor samples taken directly from patients, that if you have ACC1, you have high levels of B7H4. So we wanted to confirm that in our animal models. So we see in the ACC1 PDX tumors, we again did a very similar test where if you have high amounts of brown, you have high levels of B7H4. Our ACC type 1 PDX tumors are really positive for B7H4, whereas the ACC type 2 tumor tissue stains less so. Okay, so we've confirmed that our disease models look similar to what Dr. Ferrarotto found in the primary samples. So then we have a couple conditions that we can test. We have an untreated sort of group, and that's just looking at how do tumors grow over time if nothing has been given to them. And then we have a control. So that's a little bit like the liquid mixture that the ADC is dissolved in, because you really want to make sure that any changes in the tumor is associated because of the drug and not because of something that's sort of in the background. And then you have your B7H4 ADCs. And so the way these experiments are, you allow your tumors to grow to a certain size, and then they're divided up across the groups. They're either given control or some type of drug treatment, and then you're measuring the size of the tumors over time. So if we focus first on the ACC1 PDX models, you can see that the control and the untreated, these tumors grow bigger over time, as we would expect. But just one dose of the B7H4 ADC was able to, pretty much completely shrink away the tumors to nothing. And we saw this not only in one ACC1 model, but we saw this in a second ACC1 model, and this was very encouraging. But then in contrast, we see in the ACC type 2 model, where it doesn't have as high levels, that the tumors treated with the B7H4 ADC, that line looks very similar to the control. So those tumors were not as sensitive to the B7H4 ADC. All right, so this type of data, we often call this preclinical data, and it's absolutely essential data that needs to be generated because it's what the FDA wants to see before a drug would move into clinical trials in people. And this is something that the foundation really helps to work together with academics and companies to generate. But naturally, we want to ask, does this drug work in ACC type 1 patients? And so there are several companies that are developing B7H4 ADCs. We talked about AstraZeneca, but there's a handful of other companies, including a company called Mersana, and they all have different platforms and payloads. So that means that they're all making their antibodies a little bit different, their linkers, and their anti-cancer drugs could be different. So you could almost thinking of it as your kids playing with Legos, right? And they can create very different structures depending on the combinations of the Legos that they choose to build. So Mersana has a phase 1 trial, and again, that is looking at safety and activity of their B7H4 ADC, which is called Emi-Le or XMT-1660. They included several ACC1 patients on their trial and shared very preliminary data at one of the most famous cancer conferences that happens every year. So this is the ASCO Cancer Conference that was presented in June of this year. And so they did what's called a data cutoff time. So they looked at the data as of March 2025, and they had 9 ACC1 patients that were put on the trial. The data so far is really encouraging in terms of activity and tolerability because they saw that 5 out of 9, or 56% of the patients that were treated with the drug experienced a partial response. And that is defined as, or means that their tumor shrank greater than or equal to 30%. And so that's what we're looking at here on this chart, right? Here's in the 9 patients. Each bar represents the change in the tumor size in that patient. So if the bar goes up, that means that the tumor grew while they were on the drug. And if it goes down, that means that the tumor shrank. So you can see these are all the partial responses that were observed on the trial at that time. Okay, so then this other graph is showing you preliminary median progression-free survival. So call this PFS. This is the time at which 50% of patients remain progression-free. And this is an important readout because you could imagine that there's a drug that causes tumors to shrink, but that the tumors only shrink for a small amount of time because the tumor cells are smart enough and they figure out a way to rewire themselves so that they don't care that the drug is around anymore and they start growing again. And this is called resistance. But the preliminary result was that more than half of the patients were still on the study at the time of the data cutoff, so they weren't able to establish a PFS. So, I want to reiterate that this is very preliminary work, right? It's 9 patients that were treated, but it's exciting. This is some of the best data for any drug that has been tested in ACC patients, and so we will anxiously await Mersana to share more data publicly, and that can happen in a couple different ways. Like this cancer conference, some companies will share their data there, or they'll publish it in a paper. And so that's a role or a priority that ACCRF has, is to constantly be scanning the literature and these conferences to gather how that, when that data is shared, and then make sure that we're very quickly sharing that with the patient community. So just another plug to make sure that you are signed up for our newsletter, participate in our webinars. We share things on social media and on our website. And so this is a really important priority for us to continue to share information about Mersana, as well as if any of the other companies allow ACC patients to start recruiting to their drug. Okay, really quickly, I'm also going to go through some of the other ADC trials that are open to ACC patients. So we have 2 trials that are testing a protein, a drug that targets TROP2. We had recent data from MD Anderson over the past month showing that ACC type 2 tumors seem to have higher levels of TROP2. However, this trial does not have any inclusion criteria around TROP2 or ACC1 or ACC2. It's open to all patients. And that's because we still don't understand really whether there's a certain biomarker that will help to predict who is sensitive to this drug. That's something that will be figured out by the clinicians as the study goes on. There's a drug called enfortumab vedotin, and this is targeting a protein called Nectin-4. There's a little bit of data in the literature saying that about a third of ACCs seem to have moderate to high levels of this target, although we know that Dr. Ho, who's running this trial at Memorial Sloan Kettering, has also been working on to develop some of that data himself. And then the last trial I'd like to highlight is NN3201. This is a ADC that's targeting a protein called c-Kit, and there is a lot of data on c-Kit in ACC. c-Kit is expressed on these epithelial cells, right? And so ACC type 1 tumors that have solid histology have primarily epithelial cells, so they're very robustly positive for c-Kit. But ACC type 2 tumors with cribriform and tubular, they have a combination of epithelial and myoepithelial cells. So their epithelial cells stain positive for KIT. So we still see activity, expression for c-Kit as well in those tumors. And so that trial is open to all ACC patients. All right, so very quickly, how are we, investing in the future here, right? Because we want to always make sure that we're looking at the space. What are the new ADCs that are in clinical development, and does it make sense for those companies to test their drug in ACC patients? And so here's a couple different ways in which we're trying to invest in that space. So we can explore drug targets in ACC tumors or in individual cells. So again, it's that molecular fingerprint. Either we're taking a large chunk of tumor, right, and you get one fingerprint, or you can take that one tumor and now chop it up into individual cells. And now you have individual fingerprints that you can ask which genes or which proteins are up and down, and are any of them sitting at the cell surface based off of sort of how their sequence looks and what we know about the sequence of proteins that sit at the cell surface. We can also do things like multiplex IHC, and that's where we're looking at multiple drug targets, so sort of that brown staining, but now you have many, many different colors, and they all relate to a different protein. And you can look at them all at once on one tumor slide. And so this provides a ton of information in terms of, is your target inside the cell or is it on the surface? Is it in the tumor or is it in the microenvironment, right? Is it in the surrounding environment where the tumor kind of sits and interacts with? And not only are you asking about one target, but you're asking about how does my target relate to another target? That could be an immune cell. It could be a different target for a different drug. So this is a really powerful approach to generate a lot of information. And we have multiple projects or grants geared towards that, because everybody has a slightly different platform and a different set of ACC tumors. So here, the goal is to seed many different projects that hopefully will give us a lot of information to dig through. And then finally, we have generated something called a tissue microarray. And so what this is a slide, and it has tumor stamps from each one of our PDX models. And we can use these slides to do those tests very similar to where we were looking at B7H4, where it turned brown wherever B7H4 was. But you can do that test for really any drug target. And so let's say a company is developing an ADC against protein 123. We can take our slide and we can do that test and say, well, how does protein 123 look in ACC1 tumors, in ACC2 tumors? Maybe it's up in everything. And this is a different type of preclinical data that companies are interested in looking at, right? And so we can take that data, if we do it ourselves, along with the education around ACC, and teach the companies about that. And that's the first step to really get them thinking, well, maybe it makes sense to consider our drug for ACC patients. Okay, so I've talked quite a bit. Now we're going to get through to the question and answer portion. We've pulled some from the registration, but you can also submit your questions in the chat. As a reminder, this webinar is being recorded. Please don't submit any of your personal medical or confidential information into the public chat. And I'm going to stop sharing so that you can see our faces. Okay.
Great. Well, thank you so much to Nikki and Jeff for sharing such great in-depth information and insight into the current landscape of clinical trials and the available treatments for ACC. As Nikki said, we're going to start with a few of the more common questions that we received through the registration form. And then as time allows, we will take some of the other questions that have come up in the chat. So our first question, I'm going to give to Jeff. So we're talking a lot about clinical trials, but most of those clinical trials are based in the United States. So are non-US residents able to enroll in those clinical trials in the US? And what does the clinical trial landscape look like for people who aren't based in the US?
Yeah, we get this question a lot, and it's a very good one. I wish there were clinical trials everywhere available to all ACC patients, but the reality is most of the trials are here in the US. They are open to non-US residents, so it is possible to join them, but the barriers are high. Typically, in the US, the insurance would cover a large portion of the clinical trial. And unless a patient from overseas has insurance that would cover costs in the United States, they probably would have to cover most of the costs themselves, which could be hundreds of thousands of dollars. So it can be each case is different and each person's insurance is different. But there's often patients coming from overseas have to bear quite a high cost to participate, though they can. They will also need to travel here and find housing, and because often the trials you're having treatments periodically, and you're being measured periodically in scans, so it can be difficult. The other question that's related to that we get is, can you get onto a trial remotely? Can you consult with a doctor from overseas to see if you can get on a trial? And unfortunately, the reality is you would have to travel to the United States at least for the screening to see if you're eligible for the trial. So that likewise is a hurdle. So you'd have to travel here ahead of time and handle the costs, which can be substantial. I will say that the hope is that as a drug does better over time, if it does well in ACC in phase 1 and early phase 2 trials, then you're at a point where companies need to not only recruit a lot of patients, they need to recruit a diverse set of patients to show that the drug works in all different ethnicities and geographies. And so if you're getting into a registrational trial where a trial where the company wants to take the data to the FDA or to the European Medicines Agency or Japanese FDA or the Korean FDA or wherever, they're going to want to see data from lots of different places globally. And so those late phase 2 and phase 3 studies will open in Europe, in Asia, and hopefully other places. But so eventually, as the drugs get further along, which is really we're at the first time where that's happening in ACC, we should see greater availability of clinical trials.
Great. Thank you so much, Jeff. For our next question that we've been seeing a lot, I'll pose it to Nikki. So we know that ACC most commonly arises in the salivary gland, but it's also found in a lot of other locations: breast, trachea, eye, ear, vulva, skin, among a host of other locations. So are there treatments that are specific to the different ACC sites, or is it all the treatments available kind of treat any site regardless of where it is?
Okay, so I think that's a great question. And we'll take one step back, and I think we start with asking the molecular fingerprint that we always talk about. If we do a project, right? ACCRF was really interested in asking, for example, MIB. We know MIB is really important, and that started in looking at salivary tumors. But what about all of these other tumors? And so we've supported a lot of studies which do that molecular fingerprinting on ACCs of the breast, the skin, the lacrimal. And what we see is that those fingerprints are really similar. They have MIB. There was just a study showing that lacrimal ACC similarly has that ACC type 1 or 2 subgroup. So if you have the notch mutations or low P63, you tend to have a similar prognosis as salivary patients. And so the thinking is that these trials that are open and are going after targets that perhaps were identified mainly in salivary tumors, they will be applicable to ACC patients with any, primary origin, right? So lacrimal, breast. But I guess reinforcing that systemic therapy trials are only for patients who have actively growing either local recurrence or distant metastasis. And I would also add that oncologists oftentimes will do a biopsy of a metastasis to confirm that it's ACC, right? And so there, at that stage, you can also submit that in some cases, if there's enough material, that biopsy for things like P63 analysis or for additional sequencing. So the short answer is yes, we believe that a lot of the targets are shared in ACCs that start in the salivary and at other sites.
Great, thanks, Nikki. And sort of piggybacking off of that, Jeff, we hear from a fair number of patients, recurrent patients, who have had tumor profiling done to find sort of matched targeted therapies. What if the tumor has a genetic alteration other than MIB or Notch? Should that person then try a trial that isn't designed for specifically ACC patients, or what next steps are available there?
Yeah. So let me start by saying that many patients get tumor profiling reports, and they're all very different. They'll cover different genes, and so there's not like one report that will cover absolutely everything. And I hear from some patients, and I saw that in the chat, that sometimes those reports won't mention a MIB fusion or high levels of MIB in an ACC tumor. And people might think, oh, well, MIB isn't really there because it's not in my tumor profiling report. But as Nikki was saying before, 95% or maybe more of ACC patients have very high levels of MIB in the tumor, and that's probably the driver. So MIB is almost universal in ACC, and so that shouldn't show up in a tumor report. Notch often will show up, and then you're more likely to want to go to an ACC1 type study. But these reports can find other targets, and that's really the reason to take them. It may be that your tumor has an alteration, and there's a matched therapy that's approved by the FDA or other regulatory authorities and is incredibly active in other tumor types that have those alterations. And it may be that you should try those drugs. And that's really worth discussing with your oncologist, because the oncologist will hopefully have the expertise to say, well, your ACC, so all these ACCRF open studies webpage studies are probably relevant to you, but you've got a little quirk that is very, there is something extra in your tumor, and you ought to consider another drug that maybe doesn't work in most ACC patients, but might work in yours. And we can't tell you whether that's well, no one can really tell you which way is better, but that's definitely something to talk to your oncologist and explore it in depth.
Great. Thank you so much, Jeff. We are at time, so we understand if people need to hop off. But do we want to take one more question from the chat?
I was going to answer this question about, not 100% solid. because I think that's a good one. There's a lot of tumors that have a mixture of solid and cribriform tubular histology. And if you're asking yourself, where can I find that information? It's often going to be included in your pathology report. Okay, so I think historically, this is sort of an ongoing question. And for a long time, we sort of thought that if you had 30% more of solid histology, that might lead to a more aggressive disease track. However, I think with Renata's project, right, she's put forth this P63 marker. And if you are in ACC1, that tends to be lower. And if you're in ACC2, you tend to be higher. This P63 test, which is a lot like that B7H4 test that I talked about, right? You're looking at a cell turning brown if it has P63. That's available widely at hospitals, academic hospitals, community hospitals. And so ACCRF is funding a large global multi-institutional project to collect over 700 tumors from across the world to ask the question, is P63 a good and reliable marker to help us predict the prognosis? So to kind of place patients in one or two and provide them that information about, how their disease might progress. And I think as part of that project, what's coming out is that it may not just be ACC1 or ACC2. There may be an intermediate kind of bucket. And so that's one of the goals of this large project that we have funded, is really to explore, is it 1, 2, or is it 1, 1.5, 2, right? And what are the features of those patients? What does their disease journey look like? I think another question is, maybe your primary tumor that you had is ACC2, but does the metastasis that form, is that still ACC2, or could that shift as the tumor cells can kind of grow in your body to ACC1? And so we have that part of that project is also geared towards asking that question. If you have one patient and you're able to get a sample from their primary and their met, does it look the same in terms of ACC1 or 2 or different? And what's the frequency of that potential shift? So those are still ongoing types of questions.
Great. Thank you so much, Nikki. And again, thanks to Nikki and Jeff for sharing all this information and insight today. That takes us to just a little bit over time, so we're going to wrap up. I want to thank everyone who attended today, everybody who submitted really wonderful questions. If we didn't get to your question or you have other questions that have come up, you can email us at info@accrf.org, and we'll respond to your questions as best as possible. Also, if you have ideas for topics you'd like to see covered in future webinars, you can also email us at info@accrf.org and let us know topics that you'd like to see covered in the future. But yeah, as always, you can stay connected with us on social media and through our email list where we send out our research updates. And you can also find us on social media. So all those are available on our website, accrf.org. And we really appreciate your time and your support and your efforts. So thank you so much for being here, and we hope you enjoy the rest of the day. Thanks, everybody.
Thanks.